Welcome to the Advanced Fluorescence Microscopy and Image Processing course. So today is the lecture number 16th, and today we will learn object segmentation using ImageJ or Fiji. So let me tell you what is object segmentation. So in this image you can see there are different vegetables are there, okay, and human eye can easily distinguish them or separate them, okay. But if you ask the computer to separate the objects, okay, based on color or based on intensity, it's a very difficult job for computer. So if you ask computer to segment each of this object from this image, it will do the job like this, okay. So what human beings can easily accomplish may pose very daunting technical problem for computer software, especially when trying to separate the touching objects, okay. So object segmentation is the same thing. Segmentation refers to the process by which an image is subdivided into constituent regions or objects. And once the image is segmented, these objects can be further processed or analyzed for quantitative information. And what makes the biological images difficult to segment is that they are usually very noisy, messy, or of very irregular shapes, and segmentation can be combined with various filtering techniques to achieve higher accuracy. Okay? And there are different ways of segmentation that we will discuss in today's workshop. So let me show one example to demonstrate what is the significance of object segmentation. Okay, so let's say this is one time-lapse movie, and what we are seeing is the cell nucleus. It is stained with some fluorescent dye. Okay, and if you see this time-lapse movie, it shows the cells are dividing. You can see the more number of nuclei as the cell divides. Okay, so this time-lapse movie has total 93 time points, and we can scroll through those time points using this slide bar. Okay, so this is the time-lapse movie. Now let's say if your biological question is to count the number of nuclei per time point, to measure the fluorescence intensity of each of these nuclei, to measure the size or the area of each of these nuclei over the period of time, then you cannot do this thing manually because there are so many cells. If you go for making the region of interest manually, it will take a lot of time. Okay, so you need some kind of an automation so that this software can automatically identify the positions of the nuclei, and it can make the region of interest automatically. Okay, that is known as image segmentation. From this image, the software can automatically segment the nuclei, and then it can give you all the measurements that what is the intensity value for this nuclei, what is the area, what is the other features like size and shape and okay. So basically many biological information you can get from each of these nuclei quantitatively using this automated segmentation. So first thing is you need to segment the nuclei, and then the software does all the measurements whatever you like. Okay, so today I will show you how to use ImageJ for object segmentation. So I will show the first image. Okay, so this is the image with rice grains. Okay, I will open this image in ImageJ. Here I will go to open, and from my folder I'll open the image rice grain. Okay, so see, this is the rice.diff image. Okay, now if I have to count the number of rice grains in this sample, or if I want to measure the size of all the rice grains present in this sample, ideally I have to make the manual region of interest again each of these rice grain. Like I can just zoom it like this, and I can take this manual options of making the region of interests, and I can do it like this for each rice grain. Okay, and I can add each of this RO region of interest into this ROI manager. So this is the ROI manager. So what I can do is I can make multiple ROIs again each rice grain, and I can keep adding it to the ROI manager. And if I do show all, you can see whatever the region of interest that I have already made. Okay, so this is one way of going manual for identifying the number of rice grains in this image, or maybe if you want to measure some size or diameter or something like those kind of parameters or the intensity, that you can do. Okay, but it will take a lot of time because so many rice grains, and it will take almost 1 hour to make the region of interest against each of these rice grains. So how to do it in an automated way? So there is one image segmentation option available in this image J, and the name is find edges. Here you can see find edges. Okay, so now where it is located, it must be somewhere here in the process menu. You can see find edges. Okay, but let's say if you do not remember that where this option exists, you can just search here that you need the option of find edges. So it will show you where it is. It is in process, and I'll just say run. So that option will be open. Okay, and what I can do is I will just close this image first. I'll close this ROI manager also. I will again open the rice image. Okay, and now I'll just use that operation find edges, and you can see all the rice grains have been automatically identified. Okay, so this is one operation which can be used for image segmentation, which is known as find edges. Okay, but it only works if your background is very clear and your particles are well separated. Okay, but let's say in this case, in this image, these coins are lying here and they are overlapping with each other, right? So if you use this find edges operation with this image, it will find the edges like this. So even there are 2 coins, it will just make it as it will just segment it as a one object, single object, instead of 2 different objects. Okay, so this find edges option may not work in all cases where the objects are overlapping like this. If the objects are well separated, you can still use find edges. So now before I will show some more options for image segmentation or the object segmentation, I will briefly discuss about image filtering, that what is image filtering and why it is used. Okay, so most of the time the biological images are very noisy. Okay, and that's why this automated segmentation tools may not work precisely on those raw images, and in that case you may need to filter your image to so that the software's it will be easier for the software to identify the objects. Okay, so filtering raw images removes the noise from the raw image and make object segmentation more efficient. And image filtering is generally performed before the object segmentation. Okay, so before you go for any tools of object segmentation, you just filter your image to make it remove noise from the image. Okay, so what is image filtering? So image filtering is the process of feature enhancement prior to image analysis, and filtering is not used solely for making an image looks good, but when properly used, it can greatly enhance the efficiency and accuracy of segmentation. Okay, and image filtering is performed on pixels, and it will alter the original data embedded in the image, and that's why you need to know what you are doing. There are various algorithms used in image processing and morphological analysis, and they perform their operations on groups of pixels like 3x3 or 5x5, which is known as kernel. Okay, now before going into all this, I will show you what the image filtering means. So let's say here you can see this is the raw time-lapse movie. Okay, and here you can see there are individual molecules, fluorescent molecules, but there is a lot of noise. Okay, and sometimes we have to capture the image with a lot of noise. Let's say we want to image these molecules for a longer time, and that's why we do not want to bleach them. So we use such a low laser power so that the molecules are barely visible. Okay, and we can image them for a longer time and avoid photo bleaching. So sometimes it is possible that your image is maybe very noisy like this. Now, if I use the bandpass filtering, there is one option in the software. So using bandpass filtering, you can see I can remove the noise and only these particles are clearly visible, right? And now if I ask the software to identify these particles automatically, it will do a better job, because now there is significant difference between signal to noise between the real particles and the background. Okay, so software can precisely identify the position of these particles in this image. But if I ask the software to detect the particle from this raw image, it will mislead it. Okay, because of the lot of noise, software may make some false detections. Okay, so this is known as the image filtering. So before doing the segmentation, or before asking the software to detect the particle, we can pre-process the image to filtering to remove the noise from the image. Okay, so this process is known as filtering, and now we will discuss more and I will show you some more options of filtering. Now, how this filtering works? Okay, so let's say here I mentioned that it's a bandpass filtering. Okay, but there are several other types of filters also, such as Gaussian blur filter. Okay, now what is the function of this Gaussian blur filter? The Gaussian blur filter, as the name suggests, it is used to blur your image. Okay, now you will question that why someone wants to blur the image. Okay, so I'll come to that answer, but before that, let me show you how this Gaussian blur filter works. So what I'll do, I will try this exercise. I will open this AUPB SN40 image here in the image. So in the image sample image, this AUPB SN40 image is there. So I will open it. Okay, so this is this image, and I will go to process, filter, and Gaussian blur filter. Process, filters, and Gaussian blur filter here. Okay, I'll open this window. Okay, now I'll click on this preview. So what will happen? What are the sigma value I will put? It will blur the image like this. If I put the sigma value 1, it will be slightly blurred. If I put the sigma value 2, you see the blurriness will increase. With sigma value 5, it will become more blur. With sigma value 10, this it is even worse. Okay, so higher the sigma value I'll put, the image will become it will increase the blurriness in the image. If I use the sigma value 20, or if I use the sigma value of 30, you see image will be completely blurred. Okay, so different level of blurring you can use with your with this Gaussian blur filter. Okay, so this is the original image, and with after Gaussian blur, you can see the image is now more blurry than the original image. Now, why someone wants to blur the image? Okay, so here you can see. Let's say this was your original image captured by your microscope, and your microscope was not having proper color illumination, and because of that, you can see the center area is bright and the periphery is dark. Okay, because of uneven illumination or lack of color illumination. Okay, so now if you ask the software to automatically segment this, what are the veins or the arteries in this image, okay, it will not do a good job because of the uneven background. Okay, so what you can do is you blur this image. So the blur, after blurring, you will see only this halo of the light. Okay, and then there is an image calculator in ImageJ by which you can subtract this image from this to get the image like this. So here you can see the even illumination. Okay, so we can do this exercise here. So I'll close this image. So I'll open this rice image. Okay, so what is the idea here? This rice image, original image, has a lot of background, right? And I want to make that background dark. Okay, so what I'll do, this is a rice image. I'll use Gaussian blur filter so that only the background light is visible and not the individual rice grains. And then I will subtract this background image from this image to generate this kind of image. Okay, so let us try this. I'll go to process, filters, and minimum. Process, filters, and minimum. Okay, and I'll also keep the preview on. So you can see with this filter, I can increase the radius, and you can see the rice grains are disappearing. Okay, 6, or even I can use scope for even 8. Okay, so now the rice grains have disappeared and I can generate this image. So I will save this image as file, save as TIFF, and in processed images folder, I will save this as rice image filtered. Okay, and now what I can do is I can open the rice image again, open samples, not open, and rice image. So I will subtract this image, the filtered image, from this original image to remove the background. Okay, so there is an image calculator that you can search here and you will find it. It is in the process menu only. And in image calculator, I will define that image 1 is my raw image, right? So that is rice.tiff. Then I want to subtract. So operation subtract. Image 2 is filtered image, and create new window, and I'll press OK. So see, this is the new image of rice grains without any background. Okay, so this is how I can remove the noise from the image or image background. Okay, so that's why sometimes you may need to blur your image, and that's why all these different filters, like this time we use this minimum filter, but Gaussian blur filter can also be used, which can make your image blurry. Okay, so I'll close this now, and let us go back to this slide. Okay, now how this Gaussian blur filter works? So when we apply Gaussian blur filter, what happens? Your image is having the intensity value like this, right? Your image is nothing but it's this kind of array of different pixel intensities, right? So when we use the sigma value of 2, it uses this kernel. Okay, so kernel is so what is kernel? Kernel is a bell-shaped Gaussian. Okay, so these are the different values. So when I apply the sigma value 2, this kernel will move on my image from first pixel to the last pixel, and it will multiply the pixel value with this value. Okay, and then it will take the sum and it will put use that amount as a pixel value for first pixel of that image. Okay, so this kernel will be moved over the entire image like this, and what are the values are obtained, that will make another image, and that is your filtered image. Okay, so this is how the filtering process works. Okay, now coming to the another operation, which is known as Fourier transformation, and this is again for filtering purpose. Okay, this is not the operation for image segmentation. This is an operation for filtering the image, for removing some kind of artifacts or noise from the image. So what is a Fourier transformation? So in Fourier transformation, any image can be converted into frequency domain. Okay, so let's say this is the real image, and if you convert it, you do the Fourier transformation of this image, the Fourier transformed image will look like this. So it converted all this complex information into the frequency domain. Okay, this is a Fourier transformed image for this one. This is a Fourier transformed image for this one. Okay, so using this function of Fourier transformation, you convert your complex image into the frequency domain image. Okay, so how it works? Let's say, suppose we just opened that rice image, right? So let me open that. I'll open that rice image, and this is a real image. But if I use that function of Fourier transformation, so go to process, and this is the function FFT. FFT stands for Fourier transformation. So if I want to convert this image, this is the Fourier transformed image of this rice grains or this image. Okay, it's a different type of representation of the image data. So Fourier transformation can convert complex signal into something that is easy and straightforward. So this is a complex signal, but Fourier transformation converts it into easy and straightforward signal. Okay, and Fourier transformation does not alter the original data. So it is not altering the original data, and this Fourier transformation is again a reversible process. So let's say from this rice image, I produce this Fourier transformed image, And if I go to inverse Fourier transformation from process and FFT and inverse FFT. Okay, this is FFT and this is inverse function. So again, from that Fourier transformed image, I can generate this original image. Okay, so in this Fourier transformed image, it's a frequency domain, so I can see any noise as a pattern. Okay, so what do I mean here? I will show you. So here I've given some example that how the Fourier transformation of some real images looks like. So if this is this kind of three boxes are there, okay, the Fourier transform image will look like this. For these different letters, the Fourier transformation image will look like this. For this kind of pictures or the images, the Fourier transformation image will look like this. So any image can be converted into Fourier transform image. Okay, and Fourier transformation is reversible process. As I showed that this rice TIFF is the original image, I Fourier transformed it, and again I did the inverse Fourier transformation to generate this original image. Okay, so it's a reversible process. Now, how to take the advantage of this reversible? So here you can see that this is the original image. Okay, and you can see some kind of a streaking pattern here at the bottom, right? And we want to remove this kind of a pattern. So what we can do is, first we can convert this original image into the Fourier transformed image, and in Fourier transform image you can see this kind of pattern gives this kind of a pattern here. Okay, so what we can do, we can delete this area from the Fourier transformed image. So basically we are removing that signal from in the frequency domain, and then again we can inverse this image to FFT. We can use the inverse FFT function, inverse Fourier transformation function, to generate the original image. And from this image you can see now this kind of streaking effect is not there. Okay, so Fourier transformation is generally used to remove any kind of artifact in the image. So this is an image artifact, okay, and that you can remove using this Fourier transformation function. So this is the power of Fourier transformation. It provides you the ability to go into the frequency domain of an image and surgically deal with the artifacts without altering the truly important part of the data. And it does so by transforming complex signals into simple patterns that can be easily isolated for removal. So I will show you one exercise for this. Okay, so there is an image in your folder known as noisy face. So open that image. File, open. This is the noisy face image. So I will open this image, and you can see there is a kind of a artifact in this image, and because of that you can see all this distortion. Okay, and we want to remove this. So what we can do is, I will convert this into Fourier transform image, process, FFT. So this is the Fourier transformed image of this image, and from this experience you can get to know that all this plus signs that you are seeing in this Fourier transform image, that is because of that pattern has generated because of this pattern in the image. So what ideally we can do is we can delete each of these small plus signs from the image like this. I am not doing very precisely, but ideally you should be a little more precise in doing this. Okay, and now if I convert this back to the original image by inverse Fourier transformation option, so go to process, FFT, and inverse FFT. Okay, so see, this is after correction. So we could remove that artifact from the original image. Okay, so this is how the Fourier transformation function should be used to remove the artifacts from the image microscopic images. So let me just close this. Okay, so this is all about filtering of the image, and there are several other filtering options are also available which you can explore on your own. But the purpose of filtration of the image before image segmentation is that it will make the image more crispy and easy for the software to recognize the objects. Okay, so now let us move on to the object segmentation. So object segmentation allows the automated detection of the objects in the image, and there are many ways of object segmentation, and some of them that we will try are find edges that I already showed, then intensity thresholding, skeletonize, watershed, dilation, erosion. So these are the different segmentation options that I will show, but these are not the only options. There are several more options are also available, and segmentation is usually accomplished by several operations. Okay, so you have to use several of these operations to segment one image properly. Okay, so let us see. So this find edges operation we already discussed, that if your objects are well separated and if you identify them or make the region of interest against them, you can use the find edges function from the image and it will identify your objects. But this operation will not work if your objects are overlapping like this, because if the objects are overlapping, it will just show it as a single object, not as two different objects. So now I will show you another way of object segmentation. So the second way that we use now is known as a threshold. Okay, so based on the intensity value of this rice grains, we will try to identify the objects. Okay, so let us open this rice.tiff file. Open and rice.tiff. So this is a rice.tiff image, and see, as we know that using the find edges function, it really works well on this image. Okay, but we will try another function of object segmentation which is known as thresholding. So go to image, adjust and thresholding. Image, adjust and threshold. Okay, image, adjust, threshold. So here is that function. Okay, so it will open this window. Okay, and here you can see we want to identify the rice grains. So what I will do is I'll say that the background is dark, so it will actually identify the rice grains. Now here, by moving this threshold value, you have to make sure that all the rice grains are detected. So let's say if I go on the right, then you see it is not detecting any rice grains, right? But if I go towards left like this, it will gradually keep on identifying more and more rice grains based on the intensity. Okay, so now try to adjust this in a way so that only the rice grains are detected. The background should not be detected. Okay, so I'm going this way, this way, this way, this way, and up to this rice grain is completely detected, I can go. Okay, but you can see on the other hand, when I push it to the extreme to detect this rice grain, okay, which is on this side of the image, it also make some false detection on the other part of the image. Okay, and that is not good for the image segmentation, because it will also identify those small things as a individual object. So basically the thresholding function doesn't work well with this kind of image where the intensity values are different from one part of the image to the other part of the image. Okay, and here you can see there are different algorithms of this thresholding. Okay, now we have used the default algorithm, but there are different algorithms. Like I can use this Huang algorithm like this. So it automatically detects this many rice grains. Then I can go for use another algorithm like this. It detected little more particles or more grains. Then this is another algorithm by which even it did better job, I would say, than the other one. So one thing is you just go for one by one to identify which algorithm works best with your image. Okay, with trial and error. Okay, so see, there are so many different algorithms. Okay, and nobody can really predict which algorithm would work best for your image, but only thing you can either read up the original articles from the research paper, you find out that which algorithm people have used to segment your image, or try each of this algorithm one by one and try them all in one shot by using this option, image adjust auto threshold. Okay, so there is an option of auto threshold from image adjust. This is a threshold option, and there is a auto threshold option. Okay, so I can go for this auto threshold. It will ask me that which methods I want to try. I will say that try all methods, means try all the algorithms that we can see here. So try all, and it is white objects on the black background, yes. And okay, so it will show me the results of all the algorithms, and I have to identify that which algorithm has worked best. Okay, so this is a default algorithm. You can see the name here in a very small font. If I zoom it, you can see this is the default algorithm. This is Hong algorithm, then this Hong 2 algorithm, this is Intermodes algorithm, this is ISO data algorithm. Okay, so which algorithm has worked best, that you can see from, I would say, if I have to select from all these algorithms, I would select this one, Otsu algorithm. It seems to me that it has done the best job compared to all other algorithms. Okay, this Otsu algorithm. Okay, so what you can do is close this, and now you know that the Otsu algorithm works well. So in this thresholding window, you can select that Otsu algorithm here. Okay, for your detection. Okay, and using the Otsu algorithm, you can identify your try screens. So this is another function of image segmentation, which is known as the thresholding base. So you have to judge the image by looking at the intensity values. Okay, so how we will decide that you will go for the thresholding algorithm. So let's say again I'll open that rice image, and I will also open another image like this, muscle. Now see, this is another image, muscle.tif. Now what will happen if I'll try to segment each of this object based on the intensity thresholding. Okay, so for this kind of objects where the intensity differences is drastic, okay, this is one object which is very which is not very dark, whereas this is another object which is very dark. Okay, so in this kind of options you should not go for thresholding based segmentation. It will not work well. Okay, but for this kind of image where there is no variation in the object to object intensity value, then the thresholding may work better compared to this kind of image. Okay, see, you can just see how the thresholding will work here on this image. I will go for the default algorithm. Okay, and okay, so see, with thresholding it could detect only this many cells, and if I try to adjust it further, it will detect, but it will start merging the dark objects. Okay, like this. So the idea is, for this kind of image where there is huge difference in the intensity of the objects, you should not go for intensity based thresholding for image segmentation, but for the image where there is not a huge difference in the flow intensity of your objects, then you can go for thresholding based segmentation. So let us move on to the next type of or before that, let me just show you another thing. What if the number of objects keep changing? Okay, so for that I'll open this image, nuxic.tif. This is the one. Okay, and I already showed you this image before. I'll just increase the brightness. Okay, and you see, so in this image you are seeing the cell nucleus stained by some fluorescent dye. Okay, and this is a time-lapse movie. It has total 93 time points. Okay, and if I go through all these time points, you can see the cells are dividing, so number of nuclei are increasing. Okay, and each of these nuclei have different brightness, means different expression level of whatever the fluorescent protein is labeled. Okay, they have different size and shape. Okay, so and you want to quantify that how the number of nuclei increases over time and how their intensity values are changing, or how their size and shapes are changing. So basically what you want is, from this image you want to detect your nuclei automatically, because you cannot go for making them a region of interest manually for so many time points and for so many nuclei. Okay, so you need some kind of automation which allows you to identify the objects automatically. So what we can do is, here several steps are mentioned to measure all these things. So first we'll go to image, adjust and threshold. Image, adjust and threshold. Okay, and I will say that it's a dark background, so it will identify the objects like this. Okay, now what I have to do, I have to select the best algorithm of thresholding so that the software can automatically detect the nuclei. Okay, so I will increase the intensity like this, and then I can try different algorithms. Let us see what they have recommended here. Threshold the image and check the threshold level lower and upper value so that the nuclei are segmented properly. Okay, so we have to basically identify. So what I will do, I'll go for that auto threshold option to see that which of this algorithm works best with this image. Okay, so let me just go to that image, adjust and auto threshold option. Okay, so it will show me that all the algorithms, how they will detect, try all different nuclei. So now out of this, I would say this is the first one, second one. I think this Hong algorithm has worked well. You see, this is the real image. I will just increase the contrast. So see, most of the nuclei have been detected using this algorithm, right? Or is there any better option available? Hong 2. I think this also looks good. Triangle algorithm. You see, most of the cells have been detected well. Okay, I think this is the best, triangle. Okay, triangle has worked best. So what I will do, I will close this, and here on this image, I will ask this triangle algorithm to detect the nuclei. Okay, so now see, throughout different time points, software has detected the nuclei, and up to the last point also it is detecting it well. And also check that how that Hong2 algorithm is working. I think over the longer time points, it seems that the Hong is working better than that algorithm. Okay, so let us go with Hong algorithm. Okay, so I selected the Hong algorithm for the detection of the nuclei. Okay, and I will do apply. Hong, background is dark. and I will ask to create a new stack. Right. Okay, now what parameters I want to measure, that I will set. So I will go to analyze and set measurements. This option, analyze and set measurement, and I will tick mark the options that I want to measure. Let's say I want to measure area, I want to measure the shape descriptor, I want integrated density, that is the fluorescence intensity, then I want centroid, then I want stack position, and also I want, let's say, mean gray value, okay, average intensity, and check limit to threshold, and digits after decimal 0.2. okay, and I will say okay, and then analyze. I'll go to analyze particles, and here I will set these parameters: size 10 to infinity. So here I am defining the size of the nuclei. Say that nuclei will be roughly in the size of 10 pixels to infinity. Okay, then circularity 0.5 to 1. They will be perfectly circular or slightly lesser than that. And then show outlines, check display results, check exclude on edges, check clear results. Okay, and no need to add to, or maybe I will add to manager, overlay composite. Okay, and then I will press OK. Process all 93 images. So it is asking me that you want to process all these 93 time points. So I will say yes. So it is doing so much counting. So what it did, from the image, it gave the numbers to the nuclei. Okay, 1, 2, 3, 4, 5, 6, 7, 8, that way in the first image. Then this is second image, third image, fourth image, and that way up to 93. Now you can see each nuclei has one number. Okay, and this is the kind of Excel file it has made. Not Excel, it is a you can save it as Excel though, and it has all these measurements for all this area. So let's go to the first time point, and this is the nuclei number 1. Okay, so for one nuclei the area is this, mean intensity is this, x and y position is this, its circularity is this, integrated density, like this is the fluorescence intensity, is so this is the integrated density, and this is raw integrated density, this is slice number 1, then its solidity, round, and other parameters which are not required at this point. Okay, so it will show you all these measurements for each of these nuclei. Okay, so all these nuclei are in slice 1, but then you can see if you move down, it will show the nuclei in slice 2. So in slice 2 it is starting from 31 number. That means it could find 30 nuclei in slice 1. Then in second time point it has started from 31 up to here 60. So from 31 to 60 nuclei it finds in the second time point. So similarly, this indicates the slice number, and it shows that how many nuclei have been identified in each of these slides. Okay, you can save this as an Excel file from here. Like you can save this result as a file, save as, and you can save it as Excel, and you can further work with all these measurements. Okay, so the purpose of showing you this exercise is for this kind of images where your size and shape and number of objects keeps changing over period of time, and you want to measure all this information about their fluorescence intensity, their size, area, and all this, then you can use this kind of function for automated analysis. Okay, so I'll just close it. This exercise is over. So this is how we got the results, like each of these nuclei were identified with unique number, and we got the area, x and y, and other parameters that we already mentioned. Okay, so this is it. I will close these images. So now coming to the another feature of image segmentation or the object segmentation. So another operation which is known as skeletonize. Okay, so that is there in the process menu under the binary tab. You can see process, binary, and here is the option of skeletonize. Okay, so this function is used to generate the skeletonized structure of this filamentous structures. Okay, so this is the original image, and you can see some, let's say, neuronal cell or something, and this kind of filamentous structure is there. And if you just need 1 pixel thick lines for all these structures, then you can use the skeletonize function. So it will generate only the 1 pixel thick line for whatever your filamentous structure is. Okay, so at some point we'll use this. I will show you in the last exercise that how to use this skeletonize function. The next operation is the morphological watershed. Okay, so that is another way to perform image segmentation based on similar or shared features is morphological watershed. So how this watershed works? All grayscale images can be viewed as a topological surfaces. Okay, the lower the intensity or the darker the pixel, the deeper it gets. So all dark areas are basins that can collect water. If you start filling the basin with water, you will begin to find the ridge lines in an image. The software will mark a pixel thin line at any ridge lines and thus segmenting the object. Okay, so how it works? So let's say this is your image. Okay, and you are seeing that the dark pixels are the basins that can collect water. Okay, so this image is being represented as a topological image, and in topological image it looks like this. Where the dark pixel is there is a basin. Where the bright pixel is there, it's a surface. Okay, so if you start filling water into this basin, then when you will keep filling water, it will happen like this. So at one point, okay, you will see that this ridge line takes place. Okay, and the software will consider this as a cutoff for this structure. Okay, and it will segment the object like this. So this is known as the watershed algorithm for image segmentation. So see, this is the original image, and the software sees it like the topological surface like this. Okay, and after watershed, so if you want to segment this kind of image after watershed algorithm, you can see the software has done a nice job of identifying the object from this image, okay, using the watershed algorithm. So here is the exercise. Open image BNW cell. So file, open, and this BNW cells, right? Now you see here most of the cells are joined, okay, and that's why they will be considered as a single object, but we want to separate them. So what we'll do, we'll use this watershed algorithm to segment these cells. Okay, we'll go to process, binary, and watershed. So here you can see the watershed algorithm here. Okay, process, binary, watershed. Okay, now what it did, it actually could not identify the real cell because it considered that the cells are in black in the black signal and the background is white. But what we'll do is, before doing this, I will convert this image. I will invert this image like this. Okay, and now I'll use that watershed, process, binary, and watershed. So see, using this algorithm, it made this kind of I will open the original image so you can understand the difference. So see, this was the original image, and after applying watershed algorithm, now you can see software could make edges of these cells, and it could separate the cells like this. Okay, so this is how the watershed algorithm works. So let us move on to the other operations for image segmentation. So now let us come on to these mathematical morphology operations known as dilations and erosions. Okay, so there are many situations in which the simple thresholding or watershed or edge detection is insufficient to accomplish the task of image segmentation. So additional functions are required to perform mathematical morphology. In this process, a small shape or the structuring elements is translated across the image during the course of the processing. And this can be best illustrated in two most basic operations in mathematical morphology known as dilation and erosion. So I will show you how these functions work. Okay, so this exercise again. Open noisy fingerprint.tiff. So go to File, Open noisy fingerprint. Okay, so this is a noisy fingerprint. Okay. Apply dilation or erosion operations. So what I'll do, I'll go to, now this dilation and erosion operations are here, process, binary, and here you can see erode or dilate. Okay, so if I go for this erode, you will see all these edges are reduced in size by 1 pixel. Okay, and those small spots which are visible here, they are all gone. Okay, again go back to process, binary, and erode. Okay. So further all the lines were reduced in size by 1 pixel. Okay, now what I'll do, I'll close this, I'll open this image again, open and noisy fingerprint. Okay, now I'll use the function of dilate. So in dilate, all these edges will increase in size by 1 pixel. So let me do dilate. So you can see the lines have become thicker. Okay, if I go for a second round of dilation, you will see now the fingerprints will merge. If I go for one more round of dilation, process, binary, dilate. Okay, so the size will increase by 1 pixel. Okay. Again, let me close this and let me open it again. So in which kind of situations you will use this? So let's say this is the image and you want to remove this kind of small debris, let's say, from the image. So in this case, if you go for that erode function, go to process, binary, and just one time erode function, so that the thickness of these lines will be reduced by 1 pixel. So see, by using this erode function, I could get rid of all those spots. Okay, and now if I'll dilate this image once, go to process, binary, and dilate. So I can get the increase in size of these lines, but without those tiny spots. Okay, the debris. Okay, so this is how you can improve your image using this dilate or erode function. Okay, and there are some options. If you go to this process, binary, options, process, binary, and options, then it allows you to set some parameters for this erode or dilation. So you have to set the number of iterations. So number of iterations means the specific number of times the operation will be performed. So you want to do the dilation just for once, or let's say 5 times altogether. Okay, and the counts specifies the number of adjacent background pixels necessary before a pixel is added or removed at the edge of an object. So let's say what this count indicates. Now let's say if I'll keep adding 1 pixel line to this line, okay, then what will happen after 2 or 3 dilation, both this line will merge. Okay, I do not want that. So I'll define that only do this if there is an adjacent black pixel is available, or adjacent, let's say 2 black pixels are available, then only you perform this dilation function on this structure. Okay, that I can define. So let me show you another application of this erode and dilate function. Okay, open this beagle.tiff image. Open beagle.tiff. Now the idea is that from this image, I want to make a boundary of this structure. Okay, like this. So how I can do is, this is my original image. I will duplicate it. I will go to image and duplicate. And I will give the name beagle-1 to the duplicated image. Okay, so this is a duplicated image. So I have 2 images. This is original beagle. I will just reduce the size. Okay. And this is beagle-1 image, right? Now what I will do, I will in this image, I will reduce the size by 1 pixel. Okay, so process, binary, and erode function. I'll go to process, binary, and erode function. Okay, so now ideally the size of this object or this structure is 1 pixel less than this structure throughout the boundary. Okay, so what I can do, I can go to image calculator and subtract this image from this to get this kind of a single pixel boundary. Okay, so I'll go to image calculator. Run, and my original image is in beagle. I want to subtract beagle-1 from beagle. Okay, create a new window. Oh, sorry, I did something wrong. Let me do other way. Image calculator. And I will subtract beagle from beagle-1. Then see, yes. Okay, so now I got this single pixel thin boundary of this structure, because what I did, I duplicated this image and then I used the erode function for 1 pixel. So this structure is 1 pixel less in size compared to this structure, and then I subtracted this image from this so that I can get the image of this single line thick pixel line for this structure. Okay, so this is also one of the approach that you can use for the image segmentation. So coming to the summary of this segmentation process. So segmentation is an extremely important first step in image analysis, and it works best if you can spend some time studying what are the best features within your image you can exploit. And frequently, no one method will work ideally by itself. Okay, segmentation is usually accomplished by combining several operations. So I will show you in the next exercise that you have to combine several operations to segment the image properly. Okay, and also try various approaches. So in this until now we studied these different ways of image segmentation, like find edges, thresholding, skeletonize, watershed, dilate, erode. We also saw some of the options for image filtering. Okay, but now this is a real life application. I will show that this is an example of how we combine various segmentation techniques in practice to separate objects in the biologically related image. Okay, so this is the image muscle.tiff. Let me open that file, open, and this muscle.tiff. Okay, now you see these are the different, let's say, muscle cells, and somehow they are of different brightness, and you want to segment these cells like this. Okay, so you have to use multiple segmentation approaches to achieve this goal. Okay. And some hints here, as we mentioned before, there is no one definite way. You can segment this image through several approaches with varying degree of accuracy. Okay. So there is no only one pipeline that, okay, first I'll go for erode, then I'll go for, let's say, skeletonize, then I'll go for thresholding to. So there is no only one way to segment this image. Okay. So we have to define a pipeline for this segmentation. Let's say first we will use erode function, then we'll use the, let's say, skeletonize function, and then the third step we'll use the intensity based thresholding, and that way we can generate this kind of image segmentation. Okay, but there is not only one way. There are multiple ways of generating this kind of segmentation with varying degree of accuracy, and you can also combine filtering and segmentation function. Okay, so first you filter the image and then use segmentation functions so that it makes the life of the software easy for segmentation. So how to solve this problem? Okay, so we'll follow this different image segmentation options here. So first you go to process and find edges. Okay, so go to process and find edges. Okay, so our image looks like this. Then the second step, now see, you are seeing the two lines are very close to each other, right? And you do not want them as a two separate line. And what you want is using the Gaussian blur filter, you can make just one line out of these two. Okay, so go to process, filters, and Gaussian blur. And check mark on this preview so you can see what radius of sigma radius you should use. So this is too much, we don't want like this, so I will reduce the blurring to 5, or maybe let me go a little lower to, now still I can see two lines, right? So the idea here is I don't want to see this two line, I want to combine it in one line. Okay, so I'll blur it so that I cannot see two lines. Yeah, with a sigma value of 3, I think, but still at some point you can see two lines. So let me go to 5. Okay, so now all structures I can see as a single line thing. I cannot see double line anywhere, right? So I think this is the best value for Gaussian blur. Okay, now the third operation that I will use is the thresholding. Okay, so go to image, adjust, and threshold. Okay, and by thresholding I have to make sure that all these lines are detected. For that also I can go for different algorithms. I think mostly this algorithm looks good to me. No, this default algorithm is not good. Hong at least accept this age, all rest of the things are good. So I'll go with this algorithm. Okay, and the next step is I will go for skeletonize because now I want single pixel thin lines. Okay, so let me go to process, binary, and skeletonize. 8-bit binary image required. Okay, so actually what we did, we haven't applied the thresholding function yet. Process, adjust, sorry, image, adjust, and threshold, and here we have to apply it. So now it is an 8-bit binary image, and on this binary image I can use that skeletonize function, process binary, and skeletonize. Okay, so this is done. Okay, so this is my segmented image of the original, that muscle.tif file. Okay, so this is how, using different operations of image segmentation and filtering. So this is a filtering option, Gaussian blur, and others are thresholding, find edges, erase are the operations of image segmentation. So using multiple operations, I finally successfully segmented this image. Okay. So this is how you should try several operations to make this kind of a image segmentation. Okay, so that's it for today, and in the next lecture we will discuss about macro programming in ImageJ, then intensity measurements and colocalization. Thank you.